Welcome to this overview on how the FDA evaluates synthetic peptides. Today we'll look at why peptides like Semaglutide, Liraglutide, Lanreotide and Ganirelix require much deeper characterization than typical small molecules. These aren't simple linear chains, they're structurally sensitive and analytically complex. The FDA expects clear proof of similarity to the RLD, supported by strong data and multiple analytical perspectives. Let's walk through what that actually means in practice. Characterization is the backbone of an FDA submission for any complex peptide. It's not just routine QC, it's evidence that your synthetic version is structurally and functionally equivalent to the RLD. The FDA wants orthogonal methods that validate each attribute from multiple angles. This helps establish structural sameness, functional equivalence, and a complete analytical fingerprint. Without these elements, the FDA can't establish confidence in your product. The FDA evaluates peptides using a full 360-degree fingerprint approach. That means confirming primary sequence, higher-order structure, impurity and degradant profiles, and isomer content. They also assess aggregation behavior and functional activity. Each of these components tells part of the story. Together, they help the FDA determine whether your peptide behaves like the RLD in every critical aspect. Over time, the FDA realized synthetic peptides share many complexities with biologics. They can form multiple impurities, degrade in different ways, and adopt various conformations. As a result, the FDA now expects a biosimilar-style analytical package for complex peptides. GLP-1 peptides like Semaglutide and Liraglutide receive even closer scrutiny due to their fatty acid conjugation, which drastically changes their analytical behavior. Semaglutide is one of the most challenging peptides to characterize because of its C18 fatty diacid modification. Lipidation affects chromatographic behavior and can produce positional isomers. Oxidative degradants also impact receptor binding. The FDA expects strong evidence that the lipid is attached at lysine 26. No misacylated impurities remain, and the impurity profile matches the RLD. Confirming these features requires LC-MS/MS, QTOF, peptide mapping, stability studies, and sometimes NMR. With Liraglutide, the biggest focus is verifying the C16 fatty acid attachment and matching the peptide map to the RLD. Developers must quantify desamidation, oxidation, and dimerization, all of which affect potency. Even a small shift, say 1 to 2%, in acylated isoforms can raise FDA concerns. That's why impurity profiling, structural analysis, and stability studies are essential for a successful comparison. Lanreotide is smaller, but its cyclic structure introduces unique analytical challenges. The disulfide pairing must match the RLD exactly, because even small mispaired variants change biological activity. Multiple conformers conform, and some are only detectable with advanced techniques like ion mobility MS or 2D-LC. The FDA generally expects misfolded variants to remain below about 5%. Functional assays also help confirm correct folding and activity. Ganirelix may be a 10-amino acid peptide, but it has a surprisingly complex impurity profile. Developers must identify truncated sequences, positional isomers, and oxidation variants. High-resolution MS is essential, along with full impurity profiling and binding or potency assays. The FDA also wants clear evidence that stability trends match the RLD. Despite its size, Ganirelix requires analytical scrutiny comparable to larger peptides. When FDA reviewers analyze your data, they focus on a few key principles. First is orthogonality. Each critical attribute must be confirmed by at least 2 methods. They also compare impurity identities and levels against the RLD. Isomer resolution is another major requirement. Beyond structural data, they evaluate process consistency, functional assays, and forced degradation behavior. If your degradants diverge from the RLD, the FDA will almost always ask questions. A successful FDA submission requires deep multi-technique analysis and a clear match to the RLD's impurity profile. Functional equivalence must be demonstrated through binding and potency data. Most importantly, your characterization should tell a clear scientific story. How your peptide aligns with the RLD across all structural and functional dimensions. When done well, this becomes the foundation of your regulatory success. In the FDA's eyes, characterization isn't a checkbox, it's the heart of peptide approval. Deep analysis, matched impurity profiles, and convincing data drive confidence and reduce regulatory uncertainty. Even a well-manufactured peptide can fail without strong characterization. But with the right analytical strategy, the path to FDA acceptance becomes far smoother. Thank you for joining this overview.